So it's important to, whenever you look at this, see what are the substances on the outside and what are the substances on the inside. For example, we have integral membrane proteins. Here's an integral membrane protein that basically spans the entirety of this structure. You're going to find a portion of it on the inside and on the outside of the protein, and it's embedded within the lipid bilayer. Now, I always love to think about this because there are going to be amino acids that are found within this part of the protein. And I think that it's valuable to think about, well, what is the composition of those amino acids? You probably don't have a lot of arginines and aspartic acids. Instead, you probably have a slightly greater abundance of residues like, I don't know, like a leucine or an alanine, or yeah, those would probably be big players, making up that region because of the fact that within that region, that is very hydrophobic. So you want hydrophobic residues. So there are some proteins that span that membrane. Then there are proteins known as peripheral membrane proteins. Now, these peripheral membrane proteins, they don't span the membrane. They're basically anchored to one side of a membrane. And in this case, we've got them anchored on the interior, the cytosolic side, the inner leaflet. Now, if we jump to the other side, what we've got are these uh, oligosaccharide chains of glycoproteins. So these are proteins that have a sugar component, and this is that sugar that's basically kind of floating out there. It's almost like a bait of sorts. And it's just floating out there waiting for something else to detect it. Um, then we have lipid anchored proteins. So this is a protein that has a modification to it where it has a lipid, and basically that lipid is inserted into the lipid bilayer and that protein is kind of stuck there. Your integral proteins are kind of complicated. Um, we've got a couple of models here, and the top models are alpha helices. So these alpha helices make up this structure that basically is embedded within the membrane. And then the bottom proteins are basically a beta barrel structure. It's a beta sheet that has been contorted and twisted to make a structure that spans a membrane. So whenever it comes to integral proteins, they're typically alpha helices or beta sheets. Those beta sheets wrap up, sorry, to form uh, a beta barrel. It's kind of a cylindrical shape. So it's not or it's not exclusively alpha helices, and it's not exclusively beta sheets that form that structure for an integral protein. Now, when it comes to lipid linking of proteins or linking proteins to lipids and getting those to associate with a membrane, we have what are known as fatty acylated proteins. We have prenylated proteins, and then we have a very like a mouthful here, glycosyl phosphatidylinositol linked proteins, or GPI proteins. Now let's look at our fatty acylated proteins. Now, what a fatty acylated protein has is a fatty acid, which is indicated right here with that long fatty acid chain. Then there's a sulfur. So it's a cysteine residue that a fatty acid is added to. So CYS, cysteine. Okay, then we've got crenylated proteins where we've added, oh man, I was talking about this in the previous video, where we've got these 5 carbon structures. These 5 carbon structures kind of add up. These 5 carbon structures are used and they're put together. What they can be used for is to produce a molecule known as squalene. Squalene can then be used to put together a molecule known as cholesterol. This isn't a pro or anti-cholesterol thing, but this is instead just showing the overlap between the building blocks of cholesterol and the building blocks of lipid linking a protein and anchoring it in a membrane. Now, the amino acid that they're linked to, cysteine. So this is a C terminal S farnesyl cysteine methyl ester. So this molecule right here, let me get my eraser out. Eraser. There we go. Uh, that chain right there is what is linked to a cysteine residue. And that cysteine residue is basically made of, or sorry, that cysteine residue is where this prenylation, this farnesylation, sorry, is added. Now, these residues, or sorry, these can vary a little bit. They're not always going to be, three of these five carbon molecules. Instead, it could be four of these five carbon molecules. Our last but not least is a GPI linked protein. GPI linked proteins are super complicated. Um, but one thing to keep in mind with all of this is that this is so we have our carboxy terminus. So the C termini of our protein gets a phosphate. Now that phosphate, or on that phosphate, we're going to add a sugar right here. We're going to add another sugar. We're going to add another sugar and another sugar. And we're going to hook that up to a molecule known as inositol, which is right here. That inositol is going to have a phosphate group. And on that phosphate group, we've got a molecule with two fatty acid chains. So this is almost the same, or it's very similar to something like a triacylglycerol or a glycerophospholipid, which, wait a minute, glycosyl phosphatidylinositol. Well, one of those head groups that can end up on a glycerophospholipid is inositol. And inositol is a six carbon ring with a bunch of OH groups on it. Here is our inositol right here. The six carbon making up that ring structure, that phosphate, or sorry, that is phosphorylid. That's where we get our glycerol. But also we have this sugar linkage that gets us to our phosphate, which gets us to our protein. So these lipid linked proteins, they all have a means to communicate with other parts of or other cells, because these are going to be projected outwards and they're going to kind of send signals meaning different things. Now, here is another depiction of what that looks like. So if we go back for this GPI, I'm going to clear all my drawings here, because your takeaway from this is that cysteine is a great place for adding a palmitate or a fatty acid and adding a isoprene subunit or a farnesyl group. So this right here is a protein. That protein's free floating around in space, but not completely, because it is linked to this glycosyl phosphatidylinositol. Now, what that looks like is here is that. That's embedded within the membrane. That phosphate connects us to our inositol. Then we've got this complex sugar matrix, which is highlighted in blue. And I'm not going to draw over it because my mouse is being funky. That phosphate links us to a part of our protein. And so it's almost like that protein can kind of float around and it's present outside of the cell, but it's still anchored in the membrane. So it's still there, it's still associated. Now, these other two models are depicted down here. We've got our inner leaflet, so these GPI proteins, GPI linked proteins, are on the outside. And I misspoke a second ago. I'm sorry. The fatty acylated proteins and prenylated proteins, those are going to be found on the inner leaflet, typically. So the top two here, inner leaflet, the bottom one is an outer leaflet. And they're going to basically hold on to a protein and keep it close to the membrane. Now, biological membranes are composed of phospholipids, cholesterol, and protein. The protein lipid ratio varies from 0.23 in nerve cells, so not a lot of uh, protein in there, compared to 3.2 in protein rich mitochondrial membranes, which, if you think about the mitochondria, the powerhouse of the cell, what does that have? Well, it's got a lot of proteins associated with the electron transport chain and electron transport complexes. There are membrane associated proteins that are either going to be integral, lipid linked, lipid linked, or peripheral. Now, one of the cool things about, if we jump back for a second, right here, we've got this GPI linked protein. And oh man, that's on the outer leaflet. Just as the case with every single fatty acid or every single lipid. Here, what we see, however, that's not really the case. Um, and whenever I say that, I'm really saying it's not the case that it's such a static and unmovable situation. Instead, it's a very dynamic situation because lipid bilayers are, by nature, they are dynamic. So we can have something known as transverse diffusion. That transverse diffusion is moving something from the outside to the inside of a cell, moving something from the outside of a membrane to the inside of a membrane. And the same thing can happen, something moving from the inside to the outside. Okay, so that is going to take place very, very slowly. That's known as transverse diffusion. Lateral diffusion, on the other hand, can happen very quickly. I always kind of think about this lateral diffusion as something that I can wrap my mind around pretty easily. I think about going to a concert and you get there and there's the floor and it's general admission. So no one has a seat, everyone's standing up, and you're like, I want to get to the front. And what you can do is you can just kind of like you bump into someone, you bump into someone else, and not like aggressively or anything that like that until you get to the mosh pit. Different story. But you're moving from the back to the very front. You just kind of negotiate around and you get up there. And that's something that you can kind of do with ease. Um, Transverse diffusion, on the other hand, let's say that you wanted to spontaneously go from your floor seats to one of those really nice, like, what are they called? Box seats, a suite. Well, I mean, physically getting up there, you need like a rope or something like that to, pull yourself up. It's going to be difficult. Um, so that would be your transverse diffusion. You're fighting against a lot there because if someone sees you get a rope out at a concert and they're like, it looks like you're trying to scale the walls here, they're going to say, security, you're out of here. But that's your transverse diffusion. You're going from one side to the other side. Now, in order to speed that up, you can use enzymes to do it. Okay. So one of the things that I was talking about a little while ago was membrane fluidity. I talked about a membrane being similar to like um, a membrane being very similar to a what's the word that I'm looking for? Waterbed or something like that. It's got motion to it. Well, the melting temperature of phosphatidylcholine containing different pairs of identical fatty acid chains is shown in the table below. So essentially what this means is that you've got a polar head group, and then you've got fatty acids that are 22, 18, 16, 14, and then a monounsaturated fatty acid with 18 carbons. So with one double bond. Okay, so what we see is that whenever the number of carbons decreases from 22 to 14, the melting temperature also decreases. Okay. Then if you compare this right here, 18 carbons with no double bonds and 18 carbons with one double bond, we see our melting temperature go from 58 to negative 22. So that 18 with one double bond, that's going to be a liquid at room temperature because its melting point is negative 22 degrees Celsius. Now, lipid bilayer's overall fluidity. So I, I, I think that one of the great values of this is to think about an organism and the conditions in which it lives in. So if you think about, let's go ahead and use two extremes. Let's say you've got E. coli, just your lab strain E. coli that has an ideal growth temperature at about 37 degrees. Um, and let's say that you made that E. coli grow nothing but a 22 carbon long fatty acid with no double bonds. It grew all of the fatty acids that it had were this bihinate. What do you think that those cells are going to be like? They're going to be like a brick. They're not going to move at all. Um, there's going to be basically no membrane fluidity. Now, if we jump all the way down to our extreme, oleate. If you have E. coli, which its ideal temperature is 37 degrees, so we've got it in an environment where it's 37 degrees and it has nothing but that oleate, well, then it's going to be too liquid, too too fluid. It's not going to work that way. So that's going to present a problem. E. coli, depending on the depending on the temperature that it's going to grow at, it's going to have a balance. It's not going to have just one. So if you're growing E. coli at 37 degrees, chances are, if you had these five fatty acids to choose from, I would probably want. Ooh, I'm going to be greedy, and I'm going to say I'm going to want a mixture of those three, because what I want is my membrane to have motion to it, to be fluid. And if I have nothing but bihinate or nothing but oleate, then I'm not accomplishing that. My structure is too solid or it's too liquid. So those are problems that we've got. Now, will membranes become more fluid with longer chain fatty acids? Longer chains. Here we saw bihinate. The longer they were, the more rigid they were. So the answer to that question is no. Then let's jump ahead. What if we increase the unsaturation of the fatty acids? We add a double bond. Well, that's a good option. If we increase the temperature, well, that would also make it become more fluid. So longer chain fatty acids, not a good solution for increasing fluidity. Um, increase in unsaturation, that's a good idea. Increase the temperature, that's also a good idea. But, at some point, E. coli, for example, are going to kind of be like people. They're going to say, well, I can't grow at this specific temperature. That's way too high. Okay. Now, cholesterol in the membrane. Cholesterol essentially buffers the fluidity of a membrane to help maintain the right amount of fluidity. So, cholesterol, what it's going to do is decrease fluidity at high temperatures. It's going to increase fluidity at low temperatures. So, if you had, you know, let's go back for just a second. If you had a bunch of oleate, well, then you can make that membrane a little bit more uh sorry, oleate at oleate, you don't want any cholesterol. And then sorry, um with the other fatty acids, you might want to add some cholesterol to increase that fluidity. Now, here's some overall breakdown of different organisms and their abundance of lipids to proteins. We already talked about mitochondrial inner membrane. That's where your electron transport chain or your fatty acid breakdown is going to take place. So you need a high percentage of proteins there. Now, for the cool stuff, membrane pores or channels for transport. Okay. We have something known as, or we have the way that something gets across a membrane, moves from one side to the other, moves moves through a lipid bilayer. We have a process known as passive transport and then another known as active transport. Passive transport is diffusion through a membrane, not through a proton through a membrane, but instead through a membrane. Now, this is going to be substances that are going to pass through a pore or channel or carrier is one way that they can do it. But the most passive transport is through simple diffusion, depicted right here. Something just going from one side of a membrane to the other. Now, typically, substances that are undergoing passive transport, they're going down a concentration gradient. That means they're going from a high concentration of themselves to a low concentration. So I always like to think of a specific ion. I've got sodium ions going from one side to the other side. Well, what's most favorable is to go from an area of high concentration to low concentration. What's arguably less favorable is to go from low concentration to high concentration. That would be against the gradient. And that's a great time to bring up at transport. Now, active transport, highlighted over here, must be coupled with another reaction to make the overall delta G less than zero. Now, this is going to it's going to pump solutes against a concentration gradient. So what that means is we're going from low to high concentration. So there are a number of different types of channel proteins and carrier proteins that do this. We'll look at a couple of examples, but what I want you to first think about is kind of build a model or build how this works. Some things can move across a membrane without the help of the protein. Those things would be steroids, water, and oxygen. What are traits of those things? They're all small, except for the steroids. If something's a little bit larger than that, it can't move across the membrane on its own. Instead, it's going to need to pass through a pore or channel or a carrier. This is going to be done down a concentration gradient. So passive transport goes down a gradient, whereas active transport goes up or against the gradient. Now, these different types of active and passive transport, we have three major types. So this first model on the far left is just a simple model. It is this is called a uniport. So on the far left, that's a uniport. That is one substance or one ion being moved from one side of a membrane to the other side of the membrane. If you look at model B, we've got two substances moving in opposite directions. This is known as two substances moving in the same direction, indicated by the arrows that we have. These two substances moving in the same direction make this what's known as a sim. So sim or same. That's why then our model on the far right, we have a blue ball and a red square. The blue ball is moving in one direction from the top to the bottom, and then the red square is moving from the bottom to the top. This is what's known as an antiport. So we have a uniport, a symport, and an antiport. Now, we have a phenomenon known as secondary active transport. This is a way in which a substance is going to be transported across a membrane into basically in a way that it kind of doesn't really want to be. So to illustrate this, we have a couple of things going on. So if you look at this model in the upper left-hand corner, what we have is a substance, and that substance is going from the reduced form to the oxidized form. So if you think back to oil rig, well, the reduced form, reduce is gain. So the reduced form has electrons. It's going to release those electrons. So that's going to give off energy. So S red to S ox. So from SRED to SOX, we have energy that's released. That energy is going to move this proton from this side of the membrane to this side of the membrane. So it's almost like that's propelling it to happen. So that proton is basically going in a direction it kind of doesn't want to go to. But then we have another protein complex that is acting as a symporter. And what it is doing is it's moving lactose and a proton one side to the other side of the membrane. Okay, now, in order to get a good understanding of this, we have to process everything that's taking place. So the proton on the left, going from the bottom to the top, unfavorable, but became favorable with the combination of that oxidation reaction. Now, when we put that proton into the top half or above my membrane or above the membrane, it's in a place that, well, it's a high concentration. So then what its natural tendency is to want to go over here. It's to go from here to here. And whenever it does that, it's moving with its concentration gradient from a high concentration to a low concentration. And what that is going to do is effectively pack up this lactose and carry it with it. It's going to take that lactose with it. It's going to see it on the other side. Now, that's the way that secondary active transport works. Effectively, that's also what's happening down here. So if you look at the bottom, we've got the hydrolysis of ATP. ATP hydrolysis releases a bunch of energy. Now that bunch of energy is going to move three sodium ions to the external side of the membrane. So it's going from an area where it was a concentration of about 5 to 15 to 145 millimoles. So kind of an unfavorable move. The potassium going from a low concentration to high concentration. Oh, man, both of those things are unfavorable. So moving those sodium and potassium ions in their respective directions, it's an unfavorable combination. Then our other protein right over here, it's taking sodium ions and glucose and moving them from this side where there's a lot of sodium to this side where there's a low sodium concentration. So the movement of sodium is favorable. This glucose is more or less taking a ride. So this is one of the important parts about secondary active transport. You could look back at this hydrolysis of ATP and say, oh, well, the purpose of that was to get glucose into the cytosol, was to get glucose from the outside to the inside. It's only going to do that, though, if it's packaged or coupled with something like sodium, going from a high concentration to a low concentration. And here's another nice picture of that. We've got all these sodium ions to illustrate this. These sodium ions going from an area where there's not a lot of sodium to an area where there's a lot of sodium. Now that sodium being transported to this area where there's a lot of sodium ions, this type of protein would be considered an antiport on the left-hand side because sodium is going in one direction and potassium is going in the opposite direction. Then we have our sodium glucose symporter. This is pumping two substances in the same direction. Moving one in a favorable direction because the sodium is going in a favorable direction. The glucose, it's in a slightly less favorable direction. So I want you to know about secondary active transport. I want you to understand secondary active transport. The way that these proteins work in concert with one another is essential for understanding secondary active transport. Now, lipid vesicles at the plasma membrane, this is one of the cool things about the way that a cell works. So if we jump back all the way to here, we've got these oligosaccharide chains of glycosphingolipids. They're on the outside. How do they get there? You know, it's nice to think that, oh, well, that protein is translated and just kind of floats through the cytosol and then pokes its head through the membrane. But in order to do that, oh, what do we know about this membrane? It's hydrophobic. It doesn't really want to mess with something that's not hydrophobic. Well, there's a little bit of a detour for that. And that is through a process known as endocytosis or exocytosis. Um, a vesicle will be produced, and within that vesicle, the part of a protein that is going to end up on the outside of a cell will be packaged inside that vesicle. So these sugars, indicated here by this carbohydrates, or that term carbohydrates, what's going to happen is those are packaged inside the vesicle. That vesicle will go, and what it's going to do is it's basically going to run up against the membrane here, and it's going to merge with the membrane. And when it merges with the membrane, basically it's going to be extended outward. And these proteins with sugars on them, they're going to be displayed outside of the cell. They're going to be on the exterior of a cell or of the cell. And that's because they were initially packaged inside a vesicle. So they are going to play a big role in signal transduction. So they're going to work with membrane receptors to transduce a signal, an external stimuli, and those are going to then be able to be used for generating secondary messengers that will go on to produce a cellular response. Now, big secondary messenger examples, here we've got cyclic AMP or cyclic GMP. We have calcium ions that are tremendous secondary messengers. And then we've got this inositol 1,4,5 triphosphate, as well as diacylglycerol. So these are all examples of all examples of secondary messengers. It's important to be able to identify each one of these players and know the differences. So this 6-membered ring right here, that is inositol. And then the positions 1, 4, 5, that's where our phosphate groups are. Now, here's a nice model for insulin signaling and what it consists of. So insulin binds to a protein complex known as the insulin receptor. Now, what that receptor is going to do is it's going to bind insulin. It's going to undergo a little bit of a structural change. Whenever it binds insulin, that signal, the presence of insulin, is going to basically be amplified. And this insulin plus insulin receptor is going to lead to all of these phosphorylations right here. Those phosphorylations are going to have, and that's going to be our activated receptor, which is then going to stimulate this right here, phosphoinositide 3 kinase. That phosphoinositide 3 kinase is going to activate and put together a molecule known as phosphatidylinositol 3,4,5 triphosphate, or pt3. That pt3 is going to activate this protein complex, PDK1, which is phosphatidylinositol dependent kinase 1. This, or pt3 dependent protein kinase. That is going to take a protein known as AKT and activate it by phosphorylating it. So PDK1 activates AKT. PDK1 is activated by PIP3, or phosphatidylinositol 3,4,5 triphosphate. That phosphatidylinositol 3,4,5 triphosphate is produced by phosphoinositide 3 kinase. That phosphoinositide 3 kinase only does that because this IRS1 protein is stimulated by the receptor to amplify that signal, to basically stimulate, hey, insulin is here, let's get activated. This activated AKT goes on to activate glucose transporters and basically stimulate a response. Hey, there's a bunch of glucose around, we got to take care of it. All right, well, that'll about wrap up membranes and lipids. And if you got any questions, please let me know. Otherwise, have a good one.